Morning biologists, in today's lesson we're going to look at cell-mediated immunity. We are going to look at the cells involved and how it goes and helps combat infections within the body. Cell-mediated immunity is an immune response that does not involve antibodies but rather involves the activation of phagocytes, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines in response to an antigen. Just as with the humoral immune response, it still involves cells, but it does not involve B cells, it involves T cells instead. So these recognize infected cells and destroy them before the pathogen inside can begin to replicate and spread to other cells. So this is particularly important when dealing with viruses. So first, an antigen presenting cell detects and engulfs and digests pathogens into hundreds and thousands of antigen fragments. These fragments are then transported to the surface, making this an antigen presenting cell, where they are presented on proteins called major histocompatibility complexes. T cells become activated towards certain antigens once they encountered it being displayed on one of these major histocompatibility complexes. So after a virus or a bacteria enters a cell, it can no longer be detected by the humoral immune response. So instead, this cellular immune response must take over. To do so, a T cell will become activated by encountering an antigen of an infected cell or virus presented on that cell surface. So the way that it does this, so the T cell receptor binding to the major histocompatibility complex is the first stage. T cell receptors recognize foreign antigens. They convey the message to the nucleus to induce a response within that cell. The antigens that then bind to small peptide fragments or epitopes displayed by the major histocompatibility complex molecules to the surface of the cell. So this is where the T cell is binding on and recognizing that antigen that has been released from the pathogen. So in cell-mediated immunity, T lymphocytes respond to the cells of an organism that have been changed in some way. For example, by virus infection, by antigen presenting, or by mutation. That's the sort of thing that happens when a cell becomes cancerous. And to these cells from transplanted tissues. That is why patients who have had organ transplants need to have immunosuppressants. The cell-mediated response is particularly important against viruses and early cancers. T cells can go and differentiate into a number of different cells. The first one being T helper cell. So T helper cells will go and produce interleukins. An interleukin is a particular type of cytokine. So a cytokine is a cell signaling molecule. The interleukins made by the T helper cells stimulate the activity of B cells, which can then go and produce antibodies against the antigen which is being presented. They can differentiate into T killer cells. So T killer cells destroy the pathogen that has that particular antigen. They will go and inject a chemical called perforin, which will go and make holes in the membrane of the pathogen, causing it the insides to leak out, which kills the pathogen. You can get T memory cells. So these live for a long time and are part of the immunological memory. So the next time that they meet that particular antigen, they will start to divide rapidly in order to go and destroy whatever is producing that antigen before it can cause any damage to the body. You get T regulator cells. So these cells suppress the immune system, acting as a control and regulatory system. So the immune system doesn't get out of control. So they stop the immune response once the pathogen has become eliminated, and make sure that the body recognises self-antigens and does not set up an autoimmune response against them. So hopefully that goes through and summarises what we need to know about cell-mediated immunity. If you have any questions, please get in contact.